Hello, this is Chapter 6, Cytogenetics, Karyotypes, and Chromosome Apparations. The Human Chromosome Set. The number and appearance of chromosomes in the nucleus of an organism is an important characteristic. Chromosome analysis is a powerful and useful technique in human genetics. Here you can see the chromosome number of selected organisms. Chromosome shape. As chromosomes condense and become visible during cell division, certain structural features can be recognized. For instance, the centromere, which is a region of a chromosome to which microtubule fibers attach during cell division. The location of a centromere gives a chromosome its characteristic shape. The centromere could be located either metacentrically, submetacentrically, or acrocentrically. Metacentric means that the chromosome has a centrally placed centromere. Submetacentric means the chromosome whose centromere is placed closer to one end than the other, and acrocentric describes a chromosome whose centromere is placed very close to, but not at, one end. Human chromosomes. Replicated chromosomes at metaphase consist of sister chromatids joined by a single centromere, where the arrows are. Metaphase chromosomes. Chromosomes are identified by size, centromere location, and banding pattern. You can see on the left you have the metacentric with the centromere in the middle, submetacentric with the centromere closer to one end than the other, and then acrocentric where the centromere is located almost at one end. Now, if you notice, they all have different banding patterns, and that the short arm of a chromosome is called the P arm, and the long arm is called the Q arm. Sex chromosomes. In humans, the X and Y chromosomes that are involved in sex determination, these have different sizes and shapes. And then you have the autosomes, which are the chromosomes other than the sex chromosomes. In humans, chromosomes 1 through 22 are autosomes. Human chromosomes are analyzed by constructing a karyotype, which is a complete set of chromosomes from a cell that has been photographed during cell division and arranged into a standard sequence. Here's a picture of a karyotype. You can see they generally decrease in size as you go from 1 through 22. And then you have your X and Y chromosomes down here. In this case, there's no Y, there's only two X's, so that means that this is going to be a female individual. The karyogram, chromosome banding patterns. You can see here they have been stained, and you can see distinctive banding patterns amongst the chromosomes. Now, there's a system of naming chromosome bands which allows any region to be identified by a descriptive address. There's the chromosome number, the arm, the region, and the band. So here, for instance, if I wanted to describe this region right here, it would be chromosome number would be 1, and then the arm, it's on the Q arm, so it'd be 1Q. It's in the region, and I said this dark blue area here, let's call it that, 2. And that's kind of it. So it'd be like, let's see, 1Q, and then 1, 2. That's essentially how it's done. Oops, sorry. So, the system of naming chromosome bands, each autosome is numbered from 1 to 22. The sex chromosomes are X and Y. Within a chromosome, the short arm is P, and the long arm is Q. Each arm is divided into numbered regions. Within each region, the bands are designated by numbers. The area marked with an arrow is called, in this case, 1. Again, chromosome 1. It's on the Q arm. It's in band 2, and then it's at 4, so it's 1Q2.4. They put the band after a decimal just to kind of differentiate the band from the region. Okay, creating a karyotype. How do you do that? Well, you can see here from this diagram that there's a handful of steps involved. You draw some blood, you add some drops to a phytohemagglutinin to stimulate mitosis. You incubate it for 2 to 3 days at 37 degrees Celsius. You add a closimid to the culture for 1 to 2 hours to stop mitosis in metaphase. You then transfer the cells to a tube which contains a fixative. Then there's centrifuge to concentrate the cells. You add a low salt solution to eliminate red blood cells and swell the lymphocytes. Then you can drop the cells onto a slide, stain them with giemsa, and then you can take a look at them under the microscope and you can start to process your karyotype. Okay, so metaphase chromosomes A, so that's what they would look like when you put them on the slide or whatever, you stain them. On the right, you can see how they would be organized with the computer-assisted program where they're all pulled and kind of straightened out a little bit and then put together into the karyotype. Keep in mind that the karyotype construction and analysis are used to identify chromosome abnormalities. Okay, constructing and analyzing karyotypes. Different stains and dyes produce banding patterns specific to each chromosome. Karyotypes reveal variations in chromosomal structure and number. In 1959, there was a discovery that Down syndrome is caused by an extra copy of chromosome 21. Chromosome banding and other techniques can identify small changes in chromosomal structure. The types of information you can obtain from a karyotype include the number of chromosomes, the sex, the presence or absence of individual chromosomes, and nature and extent of large structural abnormalities. Okay, banding techniques. You can see here there's different types of staining procedures. There's 4 common types. There's G banding that you use GEMSA stain, in which you get darkly stained G bands. You have Q banding, which is uses quinacridone, that's the Q, which causes bright fluorescent bands upon exposure to ultraviolet light. It looks the same as darkly stained G bands. They're just going to fluoresce. The R banding, you're going to use GEMSA stain again, and it's going to be the reverse of the G banding that you get if you use GEMSA stain here. The difference is you're going to use some high temperature with the R banding that's going to cause them to denature, which means unravel. And then C banding, you're going to chemically treat metaphase spreads to extract DNA from the arms but not the centromeric regions of the chromosomes. You're going to stain again with GEMSA and observe with a light microscope. So you'll see the centromeric region of the constitutive heterochromatin. Okay, some chromosomal abnormalities that you may find. For instance, on chromosome 46, there'll be a deletion at the 5p region, which will cause Cri-du-Chat syndrome, which phenotypically looks like a small head, round face, low set ears. They'll have a weak cat-like cry, low brow. They'll have a broad nasal ridge and mental retardation. So just to show you a couple of these, there's a CML as well in Charcot-Marie-Tooth syndrome. You can read about those a little bit here and also in the book if you'd like. And chromosome painting is a newer technique using fluorescent dyes to generate unique patterns for each chromosome. Any nucleus can be used to make a karyotype. Lymphocytes, skin cells, cells from biopsies, and tumor cells are examples. And sampling cells before birth, there's 2 major processes that are utilized, amniocentesis and chorionic villus sampling, also known as CVS. Amniocentesis is a method of sampling the fluid surrounding the developing fetus by inserting a hollow needle and withdrawing suspended fetal cells and fluid. It's used in diagnosing fetal genetic and developmental disorders, usually performed the 16th week of pregnancy. So it looks like this. Let me see here, close up. So in amniocentesis, you get the syringe needle is inserted through the wall of the abdomen and uterus to collect a sample of amniotic fluid. The fluid contains fetal cells that can be used for prenatal, chromosomal, or biochemical analysis. So you see here a woman undergoing amniocentesis. Now for chorionic villus sampling, CVS, you have a method of sampling fetal chorionic cells by inserting a catheter through the vagina or abdominal wall into the uterus. This is used in diagnosing biochemical and cytogenetic defects in the embryo. This is usually performed the 8th or 9th week of pregnancy. It'll look like this. So a catheter is inserted in the uterus through the vagina to remove a sample of fetal tissue from the chorion. Cells in the tissue can be used for chromosomal or biochemical analysis. A woman having chorionic villus sampling is depicted here. Okay, exploring genetics, non-invasive prenatal diagnosis. Methods are being investigated to isolate fetal cells that can pass into the mother's bloodstream, placental cells, white blood cells, and mature red blood cells for genetic testing. Changes in chromosome number or chromosome structure can cause genetic disorders. There are two major types of chromosomal changes that can be detected in a karyotype. They are a change in chromosomal number or a change in chromosomal arrangement. So changes in chromosome number include polyploidy and aneuploidy. Polyploidy is a chromosomal number that is a multiple, like 3n or 4n, of the normal haploid chromosomal number, which is n. Aneuploidy is a chromosomal number that's not an exact multiple of the haploid number. Triploidy is a chromosomal number that is 3 times a haploid number, having 3 copies of all the autosomes and 3 sex chromosomes. And tetraploidy is a chromosomal number that is 4 times a haploid number, having 4 copies of all the autosomes and 4 sex chromosomes. Okay, here you can see a triploid karyotype. You literally have 3 of all of them, including 3 sex cells here. You got 3X, so clearly a female. Okay, this is an infant with triploidy. This can show the characteristic enlarged head. And keep in mind that polyploidy results when there are more than 2 complete sets of chromosomes. Now, aneuploidy involves the gain or loss of individual chromosomes. There's monosomy and trisomy. Monosomy is a condition in which one member of a chromosomal pair is missing. For instance, there's 1 less than the diploid number, or you've got 2n, which is the diploid set minus 1. So you would have 23, 46 minus 1, so you have 45 in that case. Trisomy is a condition in which one chromosome is present in 3 copies, and all others are diploid, one more than the diploid number, so it's 2n plus 1, and so you get 2 times n, which is 23, plus 1 is 47. Cases of aneuploidy can lead to non-disjunction or the failure of homologous chromosomes to separate properly during meiosis. Here's some pictures of meiosis leading to aneuploidy. You can see how it happens here if you follow these diagrams. Take a look at them closely. In non-disjunction in meiosis 1, you'll have a pair of chromosome that fails to separate properly at anaphase 1 of meiosis. 2 gametes carry both members of a chromosome pair, and 2 are missing 1 chromosome. That would be n minus 1. So let's take a look here. In meiosis 2, non-disjunction occurs in 1 cell, producing 2 cells with the normal haploid number, 1 cell with an extra chromosome, and 1 cell with a missing chromosome. The effects of monosomy and trisomy are autosomal monosomy can be lethal. It's eliminated early in development through spontaneous abortion. Some autosomal trisomies are relatively common. Most result in spontaneous abortion. 3 types can result in live birth, and that is 13, 18, and 21 trisomy. Okay, these are trisomies. Trisomies and spontaneous abortions. Out of a survey of 4,088 abortions, this is the percentage of trisomies based on chromosome number. So you can see a significant number based on chromosome 16. Trisomy 13 is Patau syndrome. You have 47 total chromosomes. You have an extra number 13. It's a lethal condition. You have 1 in 10,000. It occurs in 1 in 10,000 births. Here's an infant with trisomy 13 showing a cleft lip and palate, which is the roof of the mouth. The hand of a trisomy 13 infant showing polydactyly, which means extra fingers. Trisomy 18, which is Edwards syndrome, means you have 47 chromosomes, you have an extra number 18. A lethal condition, 1 in 11,000 births, 80% are females. You have an extra 18 there, again female. And then trisomy 21 Down syndrome, you have 47 chromosomes, you have an extra number 21. It's the only autosomal trisomy that allows survival into adulthood. Here you can see a young girl with trisomy 21 known as Down syndrome. There's another one. You can see the 3 number 21 chromosomes right here. Keep in mind that monosomy and trisomy involve the loss and gain of a single chromosome to a diploid genome. Now, what are the risks for autosomal trisomy? The causes of autosomal trisomy are unknown. Factors that have been proposed include genetic predisposition, exposure to radiation, viral infection, abnormal hormone levels. Maternal age is a leading risk factor for trisomy. 94% of non-disjunctions occur in the mother. So here you can see maternal age and trisomy 21. The risk for Down syndrome increases as the age goes up. So that's usually why they say once you get past the age of 35, your risk starts really starting to shoot up. Same thing here. You start seeing a significant increase here in trisomic conceptions. Why is maternal age a risk factor? Meiosis is not completed until ovulation, so intracellular events may increase risk of non-disjunction resulting in aneuploidy. And maternal selection, embryo-uterine interactions that normally abort abnormal embryos become less effective. So keep in mind that the age of the mother is the best known risk factor for trisomy. Excuse me. Here we go. Aneuploidy of the sex chromosomes, more common than autosomal aneuploidy and can involve both X and Y chromosomes. A balance is needed for normal development. At least one copy of the X chromosome is required for development, and increasing numbers of X and Y chromosomes cause progressively greater disturbances in phenotype and behavior. So here's Turner syndrome, 45X. So they have one less chromosome. And this, in fact, is a person that's got a single X chromosome, as you can see here. Normal females have XX. So monosomy, the X chromosome, results in female sterility. Other phenotypic characteristics happen, but otherwise generally normal. Here's twins. These are monozygotic twins. One has Turner syndrome. The twin with Turner syndrome on the left is 45X. The other twin on the right is 46XX. In this case, the condition arose through mitotic non-disjunction, which occurred after fertilization. And Klinefelter syndrome is 47XXY. Individuals, there will be males, have some fertility problems, but few additional symptoms. They'll be male generally because they'll have that Y chromosome here. Now keep in mind that pretty much as long as you have a Y chromosome, you're pretty much going to be a male, no matter how many X chromosomes you have. So here in this picture, the young man here on the left side, he's got Klinefelter syndrome. XYY syndrome, 47XYY. Affected individuals are usually taller than normal, and some, but not all, have personality disorders. Keep in mind that changes in the number of sex chromosomes has less impact than changes in autosomes. Structural alterations within chromosomes. Changes in the structure of chromosomes include deletion, duplication, translocation, and inversion. Here you can see the structural changes in chromosomes. In a deletion, part of a chromosome is lost. Here, segment F is lost. In B, duplications have a chromosomal segment repeated. Here you'll find that segment DE is duplicated. In C, it's a translocation. Parts are exchanged between chromosomes. In this example, segments of non-homologous chromosomes are exchanged. And D, you have an inversion, which is the order of chromosome segments is reversed. You can see here segment EFG is inverted. Deletions. Deletions involve loss of chromosomal material. Deletions of chromosomal segments are associated with severe genetic disorders like Cri-du-Chat syndrome and Prader-Willi syndrome. And Table 6.3 shows you some information on those and where they're deleted at. Okay, deletion in chromosome 5 and Cri-du-Chat syndrome, associated with an array of malformations, the most characteristic of which is an infant cry that resembles a meowing cat due to defects in the larynx. So you can see here chromosome 5, P arm, and the 15 band it looks like here, they all start with 15, and then in regions 2 and 3, right? Nervous system, particular gene there, and then larynx development. So deletion of part of chromosome 5 is associated with Creutzfeldt-Jakob syndrome. By comparing the region deleted with its associated phenotype, investigators have identified regions of the chromosome that carry genes involved in developing the larynx. Translocations involve exchange of chromosome parts, often produces no overt phenotypic effects, can result in genetically imbalanced and aneuploid gametes. Robertsonian translocation is translocation resulting in Down syndrome, where Robertsonian translocation makes Down syndrome a heritable genetic disease, potentially present in 1 in 3 offspring. So this is how Robertsonian translocation and Down syndrome work. Okay. Segregation of chromosomes at meiosis in a 1421 translocation carrier. 6 types of gametes are produced. When these gametes fuse with those of a normal individual, 6 types of zygotes are produced. Of these, 2, translocational carrier and normal, have a normal phenotype, 1 is Down syndrome, and 3 are lethal combinations. Keep in mind that chromosomes can lose, gain, or rearrange segments. What are some consequences of aneuploidy? Well, aneuploidy is the leading cause of reproductive failure in humans, results in miscarriages and birth defects, and is also associated with many cancers. Here you can see chromosomal abnormalities and miscarriages. Chromosomal abnormalities and miscarriages, it's showing the abnormality on the left side and what the percent frequency is on the right. Okay, the frequency of aneuploidy changes dramatically over developmental time. Between 6 to 8 weeks and 20 weeks, about 35% of spontaneous abortions are aneuploid. Around 20 weeks, the frequency falls by an order of magnitude to about 4% stillbirths. The frequency decreases again by an order of magnitude, with about 0.3% of newborns being aneuploid. Now, other forms of chromosome changes, there's uniparental disomy, which is a condition in which both copies of a chromosome are inherited from a single parent. There's copy number variation, which is a situation in which a particular gene or chromosomal region is present in multiple copies. And fragile sites appear as gaps or breaks in chromosome-specific locations. Uniparental disomy, UPD, is associated with several genetic disorders like X-linked disorders and autosomal recessive disorders like Prader-Willi syndrome and Engelmann syndrome. Now, 2 mechanisms of generating uniparental disomy. Take a look here. Uniparental disomy can be produced by several mechanisms involving non-disjunction and meiosis. Non-disjunction in the zygote or early embryo. Normally, gametes contain 1 copy of each chromosome, and fertilization produces a zygote carrying 2 copies of a chromosome, 1 derived from each parent. Non-disjunction in both parents, in which 1 gamete carries both copies of a chromosome and the other gamete is missing a copy of that chromosome. Fertilization produces a diploid zygote, but both copies of 1 chromosome are inherited from a single parent. Non-disjunction in 1 parent resulting in the loss of a chromosome can occur. This gamete fuses with a normal gamete to produce a zygote, monosomic for a chromosome. An error in the first mitotic division results in the duplication of the monosomic chromosome, producing uniparental disomy. Fragile sites appear as gaps or breaks in chromosomes. 1 fragile site in the X chromosome is associated with a common form of mental retardation in males known as Fragile X syndrome. You can see fragile sites on the X chromosome, where they're located at. The fragile sites on the human X chromosome, sites B, C, and D, are common sites and are found on almost all copies of the X chromosome. However, A, E, and F are rare sites. Expression of A is associated with Fragile X syndrome, And then I think we have B is next page here is a photograph of an X chromosome showing a fragile site where the arrow is pointing. Keep in mind some fragile sites are associated with mental retardation. Now, copy number variation. The relationship between copy number variation and the PMP22 gene and the symptoms of Charcot-Marie-Tooth syndrome. You'll see here that having more than 2 copies of this gene can cause CMT. As the copy number goes up, the symptoms become more severe. Human diseases associated with copy number variants. Here are some associated diseases with copy number variants: Alzheimer's disease, autosomal dominant adrenoleukodystrophy, Charcot-Marie-Tooth, drug metabolism, HIV/AIDS, lupus, Parkinson's disease. You've probably heard of most all of those. You can see the phenotypic effect in the gene that it occurs at. And that will end Chapter 6. Thank you for watching.